Hello lovelies, in this video we're going to be going through your OCR-A Biology Paper 1, A-Level Biology Paper 1 that we've written for 2024. Now, we know this is a big one, so please pay careful attention to the exam skills, how Dr. Upworth is interpreting the question and how she's laying out her answers.
Identify which of the following are purine bases found in DNA. So the first thing to notice is obviously it's not RNA, so we can immediately get rid of A because that contains uracil, so it's definitely not going to be that. So obviously purine bases are you can either have purine bases or pyrimidine bases, and so these are what they look like. So the difference, if you don't remember, we've got two rings for guanine and adenine, and that gives us the purine bases, and then the pyrimidine bases are all one carbon-nitrogen ring, and so that's how you can easily tell their structure apart, and purines always bind with pyrimidines, so that's why they are different, because that allows them to have that complementary base pair structure. So we need to find the two purines, which are guanine and adenine, so the answer is C. A patient's kidney function is assessed by measuring the efficiency of blood filtration through the calculation of the glomerular filtration rate, or GFR. The GFR is determined based on the rate at which the kidneys move a waste product, creatinine, from the blood. The table shows the concentration of creatinine measured in the blood and the mass of creatinine in urine after 24 hours. Give the best estimate of the GFR to the nearest whole number. Okay, so this is just a maths question, although it's obviously quite long. It's just a maths question. So if we look at the units of all of our options, it's centimetres cubed per minute. So we're going to need to make the mass that was in the urine after 24 hours per minute. So divide that to make sure we can figure out how much mass in milligrams there was per minute. So 24 hours times 60 minutes gets us the quickest way of doing it, so you don't have to do divide by 24 and then divide by 60. So if we times them together, that gets us 1,440. So then we're going to do our 1,660 divided by 1,440, and that will get us a decimal number. And already from that number and the answer on my calculator, 1.152, I can just look down my answers, and already I know it's probably going to be D, because I know I'm going to divide by 0.01, which is the same as timesing by 1,000. So that's going to get me 115 something. So it's going to be 115.27. So if you want to do the last bit, you can obviously do it to make sure, but you're not going to get to any of those other numbers at that point. And obviously the nearest whole number is 115. So the answer is D. Water can move through either the apoplast or the symplast pathway in plant tissues. Identify the row which correctly identifies the features of each pathway. So the apoplast pathway is the one that moves kind of between cell walls, so between the cell walls of cells in the space between them, and the symplast pathway is the one that goes through the cells, including through the vacuoles. It can do that as well, but it's moving through cells, through plasmodesmata, as you can see in the diagram, and moving through the cytoplasm. So these are the two pathways. So we're just figuring out which row is correct in terms of the allocation. So let's just go literally with A and go down the column. So always go down the column first to eliminate some. So water crosses the plasma membrane. It cannot be anything that has apoplast in it, because that is not true. So we've immediately eliminated A and C. Next one, water enters the vacuoles. B and D so far are correct, so we keep them both. Water moves through the plasmodesmata. That's not true for the apoplast, but it is true for the symplast. So therefore our answer is D. Even if you just check it, the space between the cells is what water moves through intercellular space. The only one that can be is apoplast, so that is correct for D. So that just confirms our correct answer, but we only need to do the first three columns to find out that D was the one that was the only one that was right. The diagram shows a cross-section of the human brain. Give the letter which correctly identifies the function of the part of the brain labelled 3. So first of all, what is this part of the brain labelled 3? It is the cerebellum. Okay, so we're going to need to know that. No point wasting time labelling the others unless you need to do it for a process of elimination, because you can't work out what 3 is. So controls conscious thought. I know that's not right because I know that's part 1. That's the kind of the higher brain functions. Judges the position of objects. It could be that one. I'm just going to double check the others. Controls homeostatic mechanisms, that's 2, because that's part of the hypothalamus and the pituitary gland. Regulates breathing rate, that's the medulla oblongata, so that's going to be 4 or around where 4 is. So it is going to be judges the position of objects, because balance and coordination is something that the cerebellum is in charge of, and controlling kind of muscle kind of contraction and things like that with fine motor skills. So it's to do with that. So it is B. The table describes the water potential of the cytoplasm of 4 plant cells arranged in a row. So you've got the plant cells numbered and then the water potential for each one in the table. Identify the statement which correctly describes one way that water will move between these cells. So one of these is correct, and it will describe the movement of water from one cell to another. So the easiest way to do this is to literally sketch it out. This is what I would literally do. Just draw yourself quickly 4 cells, number them 1 to 4, put the water potentials in, and then literally think to ourselves, okay, this is about understanding osmosis and water potential. So the thing we have to think about is, where is the water going to move from and to? So the water is always going to move from where there's a greater water potential to a lower water potential. But we have to remember that because water potential is a negative number, the highest water potential is 0, 100% water. So it's going to move from a less negative number to a more negative number. And so then all we have to do is just draw some little arrows on and figure out which water is going to move from what cell to what cell based on the negativity of the numbers. So if we look at between cell 1 and 2, 800 is the less negative number, so it's going to go from 2 to 1. It will also go from 2 to 3, and then it will also go from 3 to 4, because minus 1,000 is less negative than minus 1,250, and that's it. That's all the movement. So 2 to 1, 2 to 3, and 3 to 4, given the cells we just have in front of us. So now we just need to go through the answers and see if they're on this list. So cell 1 to cell 2, nope. Cell 2 to cell 3, yes, but one of those is wrong, so it can't be A. Cell 2 to cell 1, yes. Cell 2 to cell 3, yes. So B is probably going to be our answer, because there's only one right answer. But if you want to check the others and not just put B, then you can. Cell 4 to cell 3, no. Cell 3 to cell 4, yes. Cell 3 to cell 2, no. So it is B. But yeah, if you figured out that B was correct, then you don't really need to check the others, but it's always worth double checking in case you've made a mistake. Identify the correct order of molecules produced in the ornithine cycle. Okay, so this is a sort of think about basically, do we know what the ornithine cycle does? So it's where urea is produced from amino acids. It happens in and around the liver, and it's how we break down the amino acids and how we get urea. But it's called the ornithine cycle because it recycles ornithine. You start with ornithine at the start of the cycle, and then it regenerates it at the end, much like thinking about things that happen in the Krebs cycle and the Calvin cycle. It's a very similar thing. Cycles always end up regenerating the thing that starts in the cycle. So I know that it's got to start with amino acid because we've got to put amino acids into the cycle, and we've got to end up with ornithine because that has to be recycled, ready to start again. So that's not necessarily needing to know the whole cycle and needing to know where ammonia and citrulline come into the cycle. You don't actually need to know that to answer this question. But here's just the proof to show you that it's definitely A. You've got the amino acid coming in, then we've got the conversion to ammonia, and we get our keto acid being made. Then we have the cycle part, which obviously starts with ornithine combining with that molecule. We get citrulline, and then it gets regenerated through the cycle. We produce urea, but also we regenerate that ornithine, which then starts the cycle again. So it's definitely A. And yeah, this was just down to, do you know that amino acids go into the cycle? Do you know that the ornithine cycle regenerates ornithine? And then you can get the right answer. An athlete is warming up before a running race. Identify which statement describes how their heart rate is controlled by the cardiovascular centre. Okay, so before I even look at what options I've got here, I'm just going to think about what I'm expecting here. So if the heart rate, if we're about to run a race, we're about to do some activity, so we want to increase our heart rate. That's what warming up and stretching does. It starts stretching those muscles, which starts triggering this increase in heart rate to try and get us ready to do this exercise. So that is controlled by the sympathetic system. I always think about the parasympathetic nervous system. That para is the same front that we put in front of things like paralyzed, because obviously it's decreasing, reducing that sensation or that activity. So it's all about decreasing heart rates. Whereas the sympathetic then is the opposite. It's to do with increasing the heart rate. So immediately in the first row, I'm going to be able to get rid of two answers because they're talking about the parasympathetic system. That's not right because that would be about slowing down the heart rate. So B and D can go. Then I've got A and C. And the difference here then is the acetylcholine, the noradrenaline, and the SA node decreasing the heart rate or increasing the heart rate. Obviously, we've said we're warming up to run a race. So it's before the race, we're getting ready for activity. So it's got to be about increasing the heart rate. So even if you weren't sure about the type of neurotransmitter, that is the only answer that then makes sense because it's about increasing the heart rate and getting ready for activity. So it's got to be C. The figure below shows a plant cell and its organelles. Give the letter which identifies where in the cell cholesterol would be synthesized. So this question is just asking, do you know which organelle produces lipids? And obviously, we hopefully know that that is the smooth endoplasmic reticulum. And then it's just about identifying that on the cell diagram. So it is Q. It's not N because that is the rough endoplasmic reticulum because it has the dots on, which are the ribosomes. It's not M because that's the Golgi. But those are the things that you could easily confuse it with if you weren't looking closely enough. But it is definitely Q. So our answer is therefore B. Identify which of the following statements contains a correct description of the function of sodium ions. So that is Na plus ions. So the first one says it's involved in nervous transmission and muscle contraction. Well, that's true because obviously we need the sodium ions to depolarize the membranes. So yes. And just before we carry on, it does say statements. Okay, so statements suggests plural. And so we're looking for more than one. And it's important to get your head around that because sometimes if you don't think that and you think you're looking for only one being right, you could get in a pickle while trying to doubt yourself. Involved in the control of water levels in the body fluid, yes. And the kidney, they get transported in and out of the filtrate. Generates healthy leaves and flowers in plants, no, because that's magnesium to do with chlorophyll. Affects absorption of carbohydrates in the intestine, yes, because it's used for co-transport. So we need that molecule to do the co-transport of carbohydrates and amino acids into the small intestine. So then if we go through our options, obviously we don't want anything that's got 3 in it. So that rules out B and D. And then the A says 1 and 2 only, which obviously ignores 4, but we need, yes, it's used for co-transport, so we need 4 as well. So then therefore, our answer is C. The image shows some cells from a garlic root tip viewed using a light microscope. Identify what stage of cell division the 2 circled cells are in. Okay, so this is something that hopefully we will have practiced and we will have done this practical ourselves and we will have looked at lots of different cells in different stages of mitosis because that's clearly what we're looking at here. So the chromosomes are in the middle of these cells, especially the one towards the right-hand side. And so anything where the chromosomes are grouping in the middle, they're meeting in the middle is how I like to remember it, is metaphase. So this is kind of what I would expect you to think when you look at this image, but if you needed a bit more help to kind of figure out why it's not some of the others and do process of elimination, we can do that as well. So anaphase is the first option. It's not anaphase because what we can't see is we can't see the classic arrow shape chromosomes being pulled to the opposite poles of the cell, which is what would happen in anaphase. It's not interphase because we would not see any visible chromosomes in interphase. There's definitely some cells in this image in interphase and you can't see any visible chromosomes there. You can just see like the circle of the nucleus. And then in prophase, they wouldn't be all meeting in the middle in a very obvious kind of clump and structure like this where you can see individual kind of arms of chromosomes sticking out. They would kind of still be in that shape of the nucleus, but you would just be able to see the start of those chromosomes condensing. So these 3 cells here are kind of prophase-y. They could be prophase, they could be sort of towards the end of telophase, which is why often you don't get asked to identify these 2 stages because it's hard to see. Whereas if you look at some of the cells that I've labelled I here that are in interphase, you can definitely see a difference between those two nuclei. You can see kind of just a faded circle where all of the DNA is uncondensed and you can't see any structures whatsoever. It's just a dark circle in the middle of the cell. That's interphase, which is obviously not even a stage of cell division because it's the stage before in the cell cycle, before the cell starts to divide. So that wouldn't be a right answer anyway. And then if you look at the prophase cells that I've circled and put a P next to, you can see that there's more condensing of DNA there, that you can actually start to see kind of little worm-shaped, chromosome-shaped, sausage-shaped structures in there. And so you know something's going on. But they look very different to the cells that are in metaphase because those chromosomes are definitely all lined up kind of in a line in the middle. And so that's definitely metaphase. And so the answer is D. Students observed the underside of a pilia, or Chinese money plant, leaf under a microscope using a 1mm grid to count the number of stomata. The counts for 5 different sections of one leaf were as follows. So section 1 was 45, section 2 was 50, section 3 was 55, section 4 was 48, and section 5 was 52. Chinese money plant leaves are roughly circular in shape. The students measured the diameter of the leaf to be 6.2 centimetres. Calculate an estimate for the total number of stomata on the leaf. Okay, so again, I would just kind of sketch this out if you're not really sure what all of these words are telling you, and so that we can figure it out. So they used 5 1mm square. They took 5 measurements of the number of stomata in 5 1mm squares from around this singular leaf. And then we've got the diameter of the leaf is measured to be 6.2 centimetres. We know it's roughly circular, so this is where they're going with this to try and get you to work out the area of the leaf, because we need to estimate the total number of stomata on the whole leaf based on the counts that we've got. So we need to calculate the area of a circle, so we need to do pi r squared. This is one of those ones where they're expecting you to know this from GCSE, so you've got to have it all like at the top of your mind. You've got to do some of this maths. Now the one thing to do, because we need to do it, we have the counts done in millimetres squared. We need to make sure we convert our area of a circle into millimetres squared when we do it. So 6.2 centimetres becomes 62 millimetres. That's the diameter. We divide it by 2 to get the radius. We times that by pi and we square it. So that's the one thing to make sure you do convert before you do the pi r squared calculation. Then we've got to get the mean of the 5 different counts, okay? Because that's the reason we've repeated 5 different counts from across different areas of the leaf, is so that we can calculate a mean, and therefore, so we're saying the average was 50 stomata per millimetre squared. So we times that 50 by the area in millimetres squared of the leaf, which is pi times 31 squared. And that gets us 150,953. So the answer is C. So just making sure and being aware of making sure we convert from centimetres to millimetres before we do our calculation, and being aware of the reason why you would do 5 different sections of one leaf and why you would count it 5 times. And that's just obviously a way of repeating so you can get a mean that is representative of the average number of stomata per millimetre squared, and you just times that up by the area of the leaf as a whole. Students set up a conical flask connected to a manometer to measure the rates of respiration in some germinating seeds. They placed soda lime in the base of the conical flasks with the seeds and measured the distance moved by the liquid in the capillary tube of the manometer. The apparatus was kept in the dark. Identify the statement that correctly explains what the students would expect to see. Okay, so we're kept in the dark, so no light, so no photosynthesis, so we're measuring the rate of respiration only. The soda lime is there because it removes the carbon dioxide by absorbing it, so that means that the only thing that we're really measuring here is the rate of uptake of oxygen. So the seeds are going to be respiring, so they're using up oxygen, but the carbon dioxide they're releasing is being absorbed by the soda lime, which means that the volume of gas inside the conical flask is going to decrease, which will decrease the pressure. This causes like a suction pull on that liquid that's in the manometer, and so the water on that side of the U-bend next to the scale is going to move down. So that's how we can kind of measure the volume of oxygen that's being used up. So A says the seed's taking oxygen, yes, and the level of the manometer will move down the scale, yes. So A is our right answer. Let's check the others though. The seeds release carbon dioxide, so the level of the liquid molecule will move up. It won't because the carbon dioxide is being absorbed. The seeds take in carbon dioxide. No, they don't. There's no photosynthesis happening, so that's not going to be happening in this case. The seeds release oxygen. Definitely no, they don't. There's no release of oxygen. They release carbon dioxide. So it is definitely A, and we've checked the others. The cardiac cycle describes how blood moves through the heart in one heartbeat. Identify the row in the table that correctly identifies the pressure differences in the heart during ventricle systole. So this is a specific part of the cardiac cycle, ventricle systole, otherwise known as contraction. So when the ventricles are contracting, where's going to have the highest pressure and what the pressure difference is going to be. So this is kind of, do you know how the valves work? Because this is all about pressure differences, is how the valves are controlled. So we have to think about what valves are open and what valves are closed when the ventricles are contracting. So the valves up into the arteries, so the semilunar valves will be open and the atrioventricular valves will be closed, because we do not want blood flowing back into the atria when the ventricle contracts. So the pressure is lower in there as well because they're empty of blood. They've done their contraction, so the pressure is lower. The pressure is kind of highest in the ventricles because they are squeezing, they are contracting. And that is what has caused the semilunar valves to open, because there's lower pressure behind those valves. So this is all about, do you know which is going to be higher or lower than each other during this stage of the cardiac cycle? So let's look at A then and go down the column as always. Start with the atrial pressure, so that's the pressure in the atriums. What is going to be correct in this column? Well, we know that it's not going to be higher than the ventricle because we know we've just said the ventricles is going to be the highest. So it could be then any of the others, but it's not going to be A anymore. So we've ruled out row A. For ventricular pressure, let's go down then. Higher than the arterial pressure, yes, that's true. Higher than the atrial pressure, yes, that's true. And then arterial pressure, yes, all of those again are true. So it still could be any of the three. But then we get to the arterial pressure, so the pressure in the arteries. The only one that could be correct is the one where it is lower than the ventricles, because as we've said, the semilunar valves are open, so it must be lower than the pressure in the ventricles to push that semilunar valve open and the blood to flow into the arteries. When we get the arteries then full of blood and the ventricles have finished contracting, the valve will shut again to prevent the blood flowing backwards into the ventricles. So it definitely is then, out of these three that's left, it has to be B. The diagram shows a spirometer trace taken from a person at rest. Identify which statement describes the changes to the trace you'd expect to see if the person was exercising. Okay, so if you're exercising, your breathing rate will increase, as well as like the depth of your breathing, which is obviously your tidal volume. Both of those will increase in order to try and get more oxygen into the body so that you can obviously have increased oxygen for doing exercise, for respiration. So the first one, A, says the tidal volume would increase, which is true. And we have said that as one of the things that we'd be expecting to see. So our answer is probably going to be A, but let's just check the others. The wavelength would increase, no, because it would actually decrease because the breathing rate would increase. The vital capacity would increase, no, and you can't really increase the vital capacity. So the biggest inhale and exhale you can do. And point B would become higher than point A, no. The image shows an electron micrograph of a section of skeletal muscle. Give the correct letter for where actin is found. Okay, so out of A, B, C, and D, which one of these is where actin is found? Actin is the thin filament. So there's the thick filament, which is myosin. Thin filament is actin. It's found in both the light and the dark bands, because the light bands contain actin only, and the dark bands contain actin and myosin, which is why they're darker. So yes, Z will have actin in it because it has actin and myosin in it because it's the dark band, and X will have actin only. So sorry, Z will have it and X will have it. The W is the Z disc, so no, that's just where the end of the sarcomere is, and the H zone is the sort of gap between the actin molecules. So it's going to be C because that's the only one that has X and Z. I think this is the idea of this question was you'd think, oh, actin is only in the light filament, the thin filament is only in the light band, so you were kind of potentially going to go for only X. But it's actually in both, if you remember, because the actin has to slide over the myosin. The image shows a section of pancreas tissue. State the name of the cells labelled X. So there is a circle around a group of cells that are like an island of different cells within the majority of the tissue where the cells look different. Hopefully the clue is from the pancreas tissue. We know that there's two different types of tissue within a pancreas, and if we've got these little groups or islands of cells, they are the islets of Langerhans. Now you can say alpha or beta cells within the islets of Langerhans, but you can't just say alpha or beta cells on its own because you can't tell the difference between those two types of cells there. Then it wants us to explain why these cells in the islets of Langerhans are a part of the endocrine function of the pancreas. So we need to be able to say what the endocrine function is and how do these cells demonstrate that they are part of that function of the pancreas. So endocrine tissues monitor and release hormones into the bloodstream, so not into a duct. That's the exocrine function. And we know that the exocrine function of the pancreas is that it obviously releases digestive enzymes into the pancreatic duct. So how do these cells show that they do that feature of monitoring and releasing hormones? They monitor blood glucose levels, and then they secrete insulin and glucagon as hormones directly into the bloodstream. So just got to be clear about the direct entry to the bloodstream, and also focus on the fact that they do have receptors that monitor those blood glucose levels. So they monitor blood glucose concentration, and they release insulin and glucagon directly into the blood or bloodstream. You can just say they release hormones. You don't have to say the insulin and glucagon and name them. You can just say they release hormones directly into the blood. Describe how glucagon is used to control blood glucose concentration. In your answer, you should use appropriate technical terms spelt correctly. So hopefully we know that glucagon is used and released when blood glucose is too low. And if we're getting a describe how question, it's a long answer question, but describe how just means it wants the steps in the process. So what happens in order to trigger the glucagon release, what happens when glucagon is released, and then how does that control the blood glucose levels. So we start with the alpha cells. These have the receptors that detect the low blood glucose. They release the glucagon into the bloodstream. Then the glucagon is obviously going to travel to the target organs, so like the liver and muscle cells, and it's going to cause and trigger the increase in glucose released from these cells. So it's going to trigger things like glycogenolysis, so the breakdown of glycogen storage into glucose, and gluconeogenesis, so the production of glucose from other different types of molecules. And these predominantly both happen in the liver. So if we say where it happens, we can say that. So there is a negative feedback loop control mechanism. This means that if the blood glucose gets too high, then it prevents the glucagon from being released anymore. So we've kind of got three sections: the detection and the release of the glucagon, how the glucagon actually causes the changes in blood glucose to actually lower it, and then the control element, because it is talking about control. There are the quality of written communication marks here, so 3 key terms spelled correctly will get us an extra mark here. So we have to just think about our spelling as we go through and make sure we're using those keywords. So here's my answer. Low blood glucose levels are detected by receptors in the alpha cells in the islets of Langerhans. These then secrete glucagon into the bloodstream, which will bind to receptors on liver and muscle cells. The binding of glucagon at receptors on the liver stimulates cells to carry out glycogenolysis, which is the breakdown of glycogen to glucose, and gluconeogenesis, which is the production of glucose from other molecules, so that more glucose is then released into the bloodstream. A negative feedback mechanism means that where blood glucose levels increase enough, the secretion of glucagon will be inhibited. Okay, so I've put all my keywords that I've spelled correctly in bold, so I've definitely got at least 3 from the list. You've got the list there so you can check it, and then just make sure that obviously you've checked the spelling as well, you've got them correct. If you were unsure about using the terms glycogenolysis and gluconeogenesis, there's potential there to just explain that, as I've done with the brackets. So if you're not sure whether you're putting the right word or not, put what you mean in brackets after it, and then the examiner knows what you mean. At least then if there are marks for the description as well as the word, then you can still get the mark there. Around 340,000 people in the UK live with type 1 diabetes. Explain what causes type 1 diabetes. So type 1 diabetes is the type of diabetes where the pancreas does not produce insulin. You do not produce insulin at all. And the reason, because it wants what causes it, the reasons behind it, is that it's going to be attacked by antibodies. So it's attacked by your own immune system. So the tissue that creates or produces and secretes the insulin is being attacked as if it was a foreign particle in the body. So the immune system attacks it. It's kind of an autoimmune condition. And it can be a genetic cause. There's lots of other causes, and we're not really sure entirely how type 1 diabetes comes about all the time. But as long as we've explained what type 1 diabetes is, what causes it, and then you could give the root of the cause as well if you want to try and get those 3 marks. Type 1 diabetes is a condition where the pancreas does not produce insulin. This is because the body's own immune system destroys the islets of Langerhans. It can be a genetic condition that is inherited. Even without that last statement, because I've said that type 1 diabetes is a condition where it doesn't produce insulin, and I've said that the body's own immune system destroys the islets of Langerhans tissue, that middle or second paragraph actually gets me 2 marks, and the first one 1 mark. So I didn't even need to mention the idea of a genetic or environmental cause to get the 4 marks here. The graph shows the change in oxygen concentration in the water and blood as they flow along gill filaments. So this is a gas exchange in fish question. So we've got the water, and you can see that as we move along the gill, the concentration of oxygen in the water decreases, and as we move along the gill, the concentration of oxygen in the water increases. Use the graph to explain how this system leads to efficient gas exchange in fish. So hopefully we know this is talking about the countercurrent flow system. What does the graph show about the concentration gradient? What can we get from this graph? And it's about gas exchange, so it's about the oxygen diffusion. And the efficiency, you can see here, it's about talking about the fact that this system maximizes diffusion of oxygen across the whole length of the gill. The countercurrent flow system means that water and blood flow in opposite directions. This is a sentence that we always should be writing when we're answering these questions. Okay, that's the first sentence we should always be writing. So you can see here, obviously, as the decrease in oxygen of the water happens, the increase in oxygen of the blood happens. So oxygen is always moving from the water into the blood along the full length of the gill. So it doesn't reach equilibrium. The concentration of the oxygen in the water keeps increasing, in the blood, keeps increasing. And so that means that we have maintained the concentration gradient, because if you didn't maintain the concentration gradient, diffusion wouldn't keep happening across the full length. As the concentration of oxygen in the water decreased, you wouldn't get that. You'd get equilibrium eventually. So the countercurrent flow system, the opposite directions, means that the concentration gradient of oxygen is maintained. So oxygen always diffuses from the water into the blood along the full length of the gill. And this is a standard answer to this question. If you just have the question, explain the countercurrent flow system in fish, or explain how the countercurrent flow system makes efficient gas exchange in gills, this would be your answer. It's the same 4 marks and the same answer, regardless of whether there's a graph or not. This is what we have to say. We have to talk about the countercurrent flow system. We have to say what that means about the opposite directions, the fact that that maintains the concentration gradient, and what that means. It means that there's always a higher concentration of oxygen in water flowing next to a lower concentration of oxygen in the blood. So it always moves from the water to the blood, and that happens for the whole length of the gill filament. And that means that you've got maximised diffusion. Fish have mucus-secreting goblet cells on the tips of lamellae. The mucus contains a high concentration of glycoproteins. Describe how these cells could produce and secrete the mucus. Do not include details of protein synthesis in your answer. So we don't need to focus on transcription or translation at all in this answer. It's just about describing how the cells produce and secrete the mucus, so how the goblet cells produce and secrete the mucus. And glycoproteins, we should hopefully know, is a protein with a carbohydrate chain attached to it. So describe questions are just the steps in the process. So what happens in the right order and as much detail as you can give. So this is just about how cells produce and secrete proteins. In this case, it's the glycoproteins. There's an extra little step here. So the ribosomes in the rough endoplasmic reticulum is where the polypeptide or the protein is going to be made. Then that's got to travel to the Golgi, which is where modification of proteins happens. So in this case, adding a carbohydrate chain is the modification. So we've got to be specific about that bit of detail. And then a secretion of a protein, or in this case, a glycoprotein, is done through exocytosis. So we're going to have to describe that bit at the end of the process as well. Protein is made at ribosome on the rough endoplasmic reticulum. The protein travels in a vesicle to the Golgi body. The carbohydrate chain is attached in the Golgi to form a glycoprotein. The glycoprotein leaves the Golgi in a vesicle, which then fuses to the cell membrane. So it is secreted by exocytosis. So I've kind of done both parts of the description and used the keyword for the last point that you can see in the mark scheme, but I would recommend trying to use exocytosis if you know that that's what that last point is about. Flavobacterium is a genus of filamentous bacteria that causes bacterial gill disease in fish. Bacterial gill disease causes excess mucus to surround the gill filaments and can cause the lamellae to fuse together. Explain why fish with the disease face into the water current for increased water flow over the gills and exhibit exaggerated opercular movements. So basically, why do the fish change their behaviour to increase the water flow over the gills? So it tells you that in the question. So we are just explaining why that happens when they have the disease. So this is the equivalent for thinking about the exaggerated opercular movements. This is the equivalent of us kind of being breathless when we are ill with it or have some kind of lung issue. So why might you need to increase your breathing rate or try and in this case increase the water flow? Because you're trying to get more oxygen because gas exchange has been reduced in some way. So why do fish with the disease have a reduced gas exchange? So if you think about back to the start of the question, there's always clues in the question to help. It's where it says the lamellae can fuse together. Okay, so if they fuse together, instead of being lots of fine, thin, branching structures, they fuse together in big clumps, that's going to reduce their surface area and therefore reduce oxygen diffusion. Okay, you've got alternatives in the mark scheme to think about the mucus or this idea that if they fuse together, they're thicker, so the diffusion distance is longer. But it's got to be something that's going to affect the rate of diffusion of oxygen and therefore reduce the diffusion of oxygen that's coming in to the blood, and that's where they're trying to increase the amount of water flow over the gills to try and maximise that oxygen delivery. So I've drawn a little sketch of my kind of lamellae, my filamentous branches, and then what happens if they all stick together and form like clumps. We've reduced our surface area, just so you can kind of visualise what we're trying to talk about here. So this was about finding clues in the question, understanding why they might need to increase the water flow, and then being able to link it to the disease and link it to oxygen diffusion. So the fusing of the lamellae has reduced the surface area, so there is less oxygen diffusing into the blood. The diagram shows some chromosomes in the nucleus of a cell. Draw around one pair of homologous chromosomes in the diagram. So homologous chromosomes should be the same size and shape because they're the same chromosome, just one of them came from each gamete or each parent. So the easiest one here is to find two that look very similar in size. I'm going to pick the two smallest ones. That seems the most logical to me. If you were picking some of the others, you've got options for a large one. There's two large ones, there's two medium ones, there's two small ones. So as long as you put a ring around two that match, so in my case I did the two small ones, that will get you the mark. Explain what features would make a pair of chromosomes homologous. So features often thinks about structural or physical features. We've already said It's got to be something to do with the same size or shape that we've seen, and that's how we've identified the one for part A. But we know that what it's basically asking us to define what a homologous chromosome is with some 3 things that you can say that homologous chromosomes are, what makes them homologous. So they are the same size and shape, they are the same chromosome, but one from each gamete or one from each parent, and they will contain the same genes in the same locations. They can contain different alleles though, and obviously that means that they are not identical. So you don't get any marks for talking about them being identical because they're not, because one comes from each parent. So although they will contain the same genes, they can contain different alleles. You've got loads of things you can say for these options in the mark scheme. I'm going to go for the fact that one chromosome came from each gamete or each parent. They're going to contain the same genes in the same order, and they're going to be roughly the same or similar size. Identify what is the diploid number of chromosomes of the organism from which this cell was taken. Diploid meaning the full number of chromosomes. So this cell was taken from an organism. We're just being asked to say what the diploid number of chromosome is of that organism based on what this cell tells us. So there are 6 chromosomes, 3 pairs of homologous chromosomes, 6 chromosomes in total. So our diploid number is 6. Can't just say 3 pairs because it asks for the actual number. Draw a diagram in the circle below to represent the chromosomes that are present in the nucleus of a gamete from this organism. So this time it wants us to represent a haploid cell. So half the number of chromosomes, so it will have to be 3 in total. And at the end of meiosis, we have chromatids, okay, or individual chromosomes, but it's made of one chromatid from each homologous pair. So that means we need one large chromatid, one medium chromatid, and one small chromatid. So we're just doing one of each. So one chromatid from one chromosome of the homologous pair, because that's what happens. So in the first stage of meiosis, we separate the homologous pairs of chromosomes, and in the second stage of meiosis, we separate the chromatids from those, the sister chromatids of those chromosomes. So we end up with one chromatid from each chromosome from each homologous pair in each of the 4 gametes. So we have to make sure we know that there's 3 different chromosomes. So we've represented that with the 3 different lengths of chromatid. And you can do that with or without the centromere, that's fine. As single chromatids, they aren't always represented with the centromere. So you can do it with or without. With things like this, you just have to make your drawings really clear. So it doesn't really matter what the length is, as long as you've got one small, one medium, one large, and you can really clearly tell the difference that you've got 3 different lengths of chromosome there. Describe the structure of triglycerides and explain how their structure is related to their functions as biological molecules in organisms. So describing the structure, so the physical bonds, the molecules, what actually makes them up is the first part. We're going to get marks for that. And then it's about explaining how their structure is related to their function. So what role do they play inside organisms, and can we relate that to their structure? So when you get a question like this, feel free to draw, sketch a little diagram to remind yourself of a structure if you need to. So it's made up of 1 glycerol molecule, 3 fatty acid chains, and then each of those is joined by an ester bond. Bonds are always going to be important when we're talking about structure. So just make sure that you know the structure, you know what molecules are made of, you know the bonds that make them and the reactions that make them as well. The fatty acid chains can be saturated or unsaturated, so this is another structural element that we can mention. So they can be an energy source, they can be broken down and used in respiration. They have to be broken down to glycerol and fatty acids, so then they can enter as respiratory substrates, so we have to break those ester bonds first to do that. They can also be an energy store because they're insoluble, so they can be stored in cells, they're not going to move between cells, and they're not going to affect the water potential of cells, importantly. And they can also act as insulators. So within the body, they can form layers of adipose tissue, and they're poor conductors of heat and electricity, so they can act as insulators in that way. So all of these things, we've got 6 marks, so there's 3 kind of marks for structure, 3 marks for the functions that's linked to the structure. So I'm going to do it that way. If you weren't too sure about whether you get all the marks for the structural marks, Then talking more about the functions, you've got options there, and there's lots of options on the mark scheme that you can talk about that then means that you can get some extra marks for the functions as well. Okay, so here's my answer. Triglycerides are made up of a glycerol molecule and 3 fatty acid chains. Each fatty acid chain is attached to the glycerol by an ester bond. Triglycerides can contain saturated or unsaturated fatty acids. Triglyceride structure makes them a good energy source, as the ester bond can be hydrolyzed during respiration, and both the glycerol molecules and fatty acids can be used as respiratory substrates. When not used to provide energy, the triglycerides can easily be stored as they are insoluble, so they do not affect the water potential of cells. In the body, triglycerides can act as insulators, for example in layers of adipose tissue around organs, to help the body retain heat energy. This is because they are poor conductors of heat. The fluid mosaic model is used to characterize the plasma membranes found in all organisms. Describe the fluid mosaic model. So what is it? What is the fluid mosaic model? Why do we have the words fluid and mosaic? Why do we use them to describe it? So what does that actually mean? So the reason we use the word fluid is because it describes the fact that the phospholipids are able to move around within the bilayer. And the mosaic part comes from this idea that there's proteins studied randomly throughout the membrane. So we've got this idea of a mosaic pattern of proteins within the bilayer. So it's a describe question. We just need those 2 points. So describing what it is. So it's a phospholipid bilayer. The phospholipids are free to move within the bilayer, which is why it's described as fluid. And there's proteins studied or randomly placed throughout the bilayer, and that's where the mosaic term comes from. This is a common question, and being able to describe the fluid mosaic model is something we have to be able to do. So this is a good way of kind of making sure your language and the way you describe this is accurate. So the membrane is described as fluid because the phospholipids are free to move, and the mosaic structure because of the proteins spread throughout the phospholipid bilayer. You'll notice the phospholipid bilayer is underlined in the mark scheme. It doesn't actually matter whether you put it in for the first point or the second point, but as long as you've used that phrase, because that is how the majority of the membrane, the structure of the membrane is formed. Betalans are water-soluble pigments present in the vacuoles of plants. The most common is betanin, which can be found in red beetroots. The structure of betanin is shown below. Explain why betanin cannot leave the vacuole. So this is really why it can't cross the membrane of the vacuole. Okay, so got the diagram here. We've got some information about it in the top of the question. So we need to use that or just use our knowledge. So this is something that whether or not they've given us a diagram and whether or not they've given us information about this, you can just use the knowledge you've got as to why some molecules can't cross the plasma membrane. So can't move through the plasma membrane. So it tells you it's water soluble. That tells you that it's charged or polar. You can see some charges, positive and negative, on the diagram as well. So that tells you that it's hydrophilic, it's charged, it's polar, it's able to dissolve in water. Therefore, it's not lipid soluble, which means it will be repelled by the hydrophobic sections of the phospholipid bilayer. So it can't just go through. Other than being charged or polar molecule, which is some properties that stop molecules from being able to move through the phospholipid bilayer, the other reason is that molecules can be too large. So if you're too large, you can't just diffuse through. So we only need one of these for 1 mark, so I'm going to go with because it is a charged molecule. A student carried out an investigation into the effect of ethanol on the permeability of plasma membranes in beetroot cells. They used the following method. They used a cork borer to cut out cylinders of beetroot. They rinsed the cylinders in distilled water. They used the serial dilution method to produce different concentrations of ethanol. They placed the beetroot cylinder in 100 millilitres of each concentration of ethanol in test tubes in a water bath at a controlled temperature for 20 minutes. They then transferred the solution from each test tube into a cuvette and measured the absorbance using a colorimeter. So hopefully we're happy that this is the membrane permeability practical. It's all going to be about the practical skills related to this practical that hopefully you will have done or seen done. So with practical questions, I always try and think, okay, make sure you know what the independent variable is, what's the dependent variable, and kind of maybe think about what you think the outcome or what you predict the outcome would be, knowing what you've done with this practical if you've done it before. So the independent variable is the concentration of ethanol, which is a solvent. So this is a solvent version of this practical because you can do temperature, you can do different versions to see what factors are going to affect the membrane permeability. In this case, we're using a solvent. The dependent variable, what we're measuring, is the absorbance. So we're measuring the absorbance with a colorimeter. So that means to me, I'm thinking, okay, so more pigment leaching out would mean a higher absorbance. And that is what I'm trying to make sure I'm thinking about when I'm going to then following answer any questions that are going to come up based around this. The student is provided with a stock solution of 95% ethanol. Using calculations, show how they would produce the 100 millilitres of the 20% ethanol solution needed for the investigation. Okay, so this is just a serial dilution question, or a question about dilution and calculating how much you would need to dilute a solution to get it to be the right concentration. So for any question like this, we're going to use a nice easy method that I always go to because I find these questions sometimes can be quite hard to get your head around. So we're going to use the C1 times V1 equals C2 times V2 method. So C1 is the start concentration, V1 is the start volume, so that's sort of like the stock solution and how much you make it up with. C2 is the final concentration and V2 is the final volume. So what volume do you want to end up with at the end and what concentration do you want to end up with at the end? What solution are you making? So we've got the 20% ethanol solution and 100 mils of it. That's our final concentration and final volume. We know the concentration at the start, which is the stock solution, that's 95%. So all we need is we need to figure out what the volume of that stock solution we need to make up our 20% solution to start with, and then we will need to obviously dilute that with water. So we need to rearrange for V1, which means we get C2 times V2 divided by C1. So now we just need to put those numbers into that equation, and then hopefully we'll find V1, and then from that we can find out how much water we would need to add. So when you're working with percentages in this equation, we should use decimals. So V1 equals 0.2 times 100 divided by 0.95. That gets us 21.05 millilitres of stock solution. So that's how much stock solution you would need to take the volume you need to put into a new tube or a beaker, and then now you need to obviously make that up to 100 mils with the distilled water. So we would obviously need to just work out how many to add. You'd obviously, if you were a chemist, you could have done this in a volumetric flask, 100 ml volumetric flask, because there's a meniscus line on there where you can make it up to exactly 100 mils. Or for this question, we need 2 marks, so we need to say the volume of distilled water we're going to add to our 21.05 mils of stock ethanol, and then that will give us 100 millilitres of 20% ethanol solution. In the space provided, plot a graph using the data in the table. So we've got the table that shows the results, we've got the ethanol concentration percentage, and then we've got absorbance in arbitrary units as well, and that's the mean absorbance from those repeats. Okay, so axes first. So we're going to have ethanol concentration on the x-axis, and that's going to be our percentages. When you're doing these graphs in the exam, most of the time, the axis should be very simple, and the space they've given you to draw the graph will fit the graph that you need to draw and will fit the axes on if you're using a sensible linear scale. So there shouldn't be not enough space or not enough space to draw the graph, and it should mostly fill the space you've been given. So you shouldn't end up with a tiny graph in a corner, and you shouldn't have to end up needing to like extend your axes or anything like that. If you're doing that, you've probably picked a weird scale or you're doing something wrong. So absorbance is going up the y-axis, and it's just arbitrary units. So for this one, I've got to go from 0.02 to 0.6, nearly 0.7. So luckily, if you go up this in 0.1s, obviously each one of these tiny squares will then be 0.02. So that is perfect for what we need it for. And we can get all the way up to 0.7, which is just above where we need it to go for. I hadn't actually put in my axis lines, so I've done that now, and obviously you should do that with a ruler. So now we've just got to plot our points Obviously, we need to do this accurately. So that first one, as we said, is 0.02 because one of those squares is worth 0.02 because we're dividing 0.1 by 5. So that tells us what each of the tiny squares is worth. So that goes with the 20 concentration. Then obviously 0.3, we're just going to go in between the next two squares. You only get tolerance of half a square. So that is the worth doing, making sure you're really accurate with plotting these points. Otherwise, you may lose marks. So then we have 0.06, so we just need to go up 3 tiny squares. Then we're going all the way up to 0.66, so we need to go 3 tiny squares up from 0.6, and then we've got, again, just half a square up for the 100% concentration. And then we just do our line of best fit. I've done a smooth curve through these points. That is how best to do this data. And then I'm just going to go back and double check I've done all of the things that I need to get marks. Got my axis titles. I've included the units in brackets. Make sure you don't forget that. Just copy exactly what is in the table title for your x-axis and y-axis. Do exactly as it is in there with the units. I've got my linear scales, which work and they're fine. I've not accidentally skipped a number or something. I've plotted my points. I'm happy that they're accurate. I've done crosses, which is very important, so that the examiner can see exactly where the middle of my cross point is, exactly where I've drawn those marks, even when my line is drawn through them. If you do dots with a pen or a pencil, one, you can't tell clearly where the middle of the dot is, and you may not get it accurate, especially if you're trying to make them bigger. And then when you draw a line of best fit through them, those dots can completely disappear and be really hard to see. So try and make sure you use crosses at all times, and then your line of best fit should go through the middle-ish of those crosses. It's not going to be 100% perfect, and mine's obviously a little bit wobbly, but it's plotting the pattern that the data is showing, and that's what's most important about your line of best fit. Explain the pattern shown by the graph. So we need reasons to explain why this pattern has happened, and the pattern that we've got is that as we increase the absorption, so as we've increased the ethanol concentration, the absorption has increased. What does that actually mean? Well, it means more pigment has leached out of our cells. Why has more pigment leached out of our cells? Well, it's because the membrane has been made more permeable by that ethanol concentration. So we've got to link the increase in ethanol concentration to the increase in absorbance, and that is linked by the fact that there's more pigment leaching out because the ethanol, the increase in concentration of ethanol, has made the membrane more permeable. And that's what it wants to see here. It wants to see that link, that you understand what the ethanol has done, and that you understand that idea of being more permeable of a membrane means more pigment will leach out, which gives that higher reading on the colorimeter because there's more pigment in that solution for the light to try and pass through. As the concentration of ethanol increased, it disrupts the phospholipid bilayer, making the membrane more permeable. This causes more pigment to leak out of the cells, which increases the absorbance. So I've written mine maybe put it back to front from the mark scheme. So I think that makes more sense to me to say as the ethanol concentration increases, then the absorbance increases. That would be the description of the pattern. So I've used that as to frame my explanation. So as the concentration of ethanol increases, what does it do? And why does this cause the increase in absorbance? Because more pigment leaches out of the cells. So hopefully that makes sense, and that shows you how it's kind of the easy way of thinking about how to get those 2 marks. Hand sanitizer gels work because they contain ethanol. Effective hand sanitizers should contain a minimum of 60% ethanol, but most contain concentrations higher than 80%. Use the data from the table to explain why. So we need to explain reasons why. So think about what hand sanitizer gels do. They kill bacteria. Okay, so effective hand sanitizers are going to kill bacteria and they will be working the best. So we need to link our information we've got from the table about the 60% and 80% ethanol and why that might mean that an 80% ethanol hand sanitizer is better than a 60% ethanol hand sanitizer. And obviously we have to understand and show that we need to link that to killing bacteria. And this question is all about membrane disruption. So we know that the ethanol is disrupting membranes. So disruption of those bacteria membranes is going to cause them to burst or die, absorb nutrients, whatever it is that's going to cause them to break those cells and damage them. So if we look at the table, we've obviously got the low absorbance at 60, but the much, much higher absorbance at 80 and 100. So The absorbance we have linked, and we've just explained in that question beforehand, that greater absorbance is because of more membrane disruption and therefore more substances leaking out of the cells. So high membrane disruption is going to be more effective at killing bacteria because it's going to disrupt the membranes more and therefore increase the chance of those bacteria dying because all their insides are going to come out through the membrane. So below 60% ethanol, there's very little disruption to the membrane as only a small amount of pigment was released. So that's us using information from the table. At 80%, there was a lot of pigment released, showing it would be more effective. Hand sanitizers with less than 60% would not disrupt the membrane of bacteria enough to kill them. And you could say might, you could say would, or you could flip that around, as you can see in the mark scheme, and you could explain and start talking about the fact that why 80% would be better. So you could link that to that idea that if you want an effective hand sanitizer that kills a lot of bacteria, if you use a higher concentration of ethanol, more damage to the membrane will be done. ATP production occurs in both photosynthesis and aerobic respiration. Complete the table below to show which statements describe each process. For each statement, place a tick in one column, both columns, or not at all. So the first one, it says electrons pass between protein carriers. So this is just the electron transport chain. So both photosynthesis and respiration have an electron transport chain, so we need to tick in both. Next row says electrons are excited by light energy. Only photosynthesis involves light energy, so then we're just going to tick the photosynthesis box. The next one says it involves chemiosmosis. So this is just creating that chemical gradient of H+ ions, which is then used to make ATP through ATP synthase. That's what chemiosmosis is about. Both processes produce ATP in this way. And this is all about ATP production in total between the two. So yes, the production of ATP in both involves chemiosmosis. Includes photophosphorylation. So again, the word photo here is suggesting of light. Light energy is what is used to drive that phosphorylation, which helps to produce ATP in photosynthesis. And so yes, this is just photosynthesis, it's not respiration. Oxygen being the final electron acceptor, that means oxygen is needed or used in the reaction, and we should know that oxygen is the final electron acceptor at the end of the electron transport chain in respiration only. Okay, we don't need oxygen, we don't put in oxygen into the process of photosynthesis, it produces oxygen at the end. So that is respiration only. And then lastly, involves the matrix and the cristae. So these are both structures we should recognise as being in mitochondria. And obviously the only process here that uses mitochondria or takes place in the mitochondria is respiration. Okay, so you get a mark for each column being correct. So it does mean that you could potentially have ticked some of them wrong, say for respiration, but if the photosynthesis column is all correct, then you can have one mark. The Krebs cycle is needed to produce a large amount of ATP. State where the Krebs cycle occurs. So we don't need to do anything about this diagram for now because it hasn't asked us to look at anything, we just know that it's a diagram of the Krebs cycle. So where's the location where this stage of respiration happens? We should hopefully know it takes place in the mitochondria, but specifically the matrix. You aren't going to get away with saying mitochondria as your answer on its own because we are now at A-level stage. We know a bit more detail about this. We know exactly where in the mitochondria the Krebs cycle takes place. It takes place in the matrix. So if you just put matrix, you get a mark. If you just put mitochondria, though, you do not get a mark. Give the name of the molecule or molecules represented by the letters in the diagram. So now we're looking at this diagram properly. We've got a diagram of the Krebs cycle. Obviously some bits are missing. They're represented by letters. We have to figure out which thing goes in these gaps where the letters are, and it could be more than one molecule. So the first part is something that's coming into the Krebs cycle. So what's coming in at that start point, it's acetyl-CoA or acetyl coenzyme A, which we hopefully know comes from the link reaction. So for B and C, they're both at the point where there's a loss of carbon. Okay, so there's a loss of carbon from 6 carbons to 5 and then 5 carbons to 4. This is the stage of the cycle that we call decarboxylation. So carbons are being removed, and this is the point then where those carbons then end up being carbon dioxide, and this is where the carbon dioxide from respiration is being released. And then finally D, we've got an arrow from D to ATP, so it's whatever is in D is being used to make or produce that ATP. So what is ATP made of? It's made of ADP and inorganic phosphate, or PI. So this is why they said molecules in the question, because obviously that's 2 as opposed to all the others being just 1. So acetyl-CoA for A, B and C are both carbon dioxide, and then D is ADP and PI, or inorganic phosphate. The rate at which the Krebs cycle proceeds is carefully controlled. The rate of the enzyme isocitrate dehydrogenase 3, or IDH3, which produces the first reduced NAD in the Krebs cycle, so we're here between our 6 carbon and our 5 carbon molecule. This is reduced, the rate of the enzyme is reduced as ATP begins to accumulate. Okay, so I would just take this bit out and write myself a little note to make sure I understand what the bit of information is telling me. The more ATP is made, or when ATP accumulates or builds up, that slows down or reduces the rate of the enzyme-controlled reaction controlled by enzyme IDH3. So it slows down the rate of reaction of that enzyme. So that stage of the Krebs cycle is then reduced. Explain how this is an example of end product inhibition. So with an explain how question, we need to be saying how is this happening, and it specifically will say how does this show, or how is this an example of end product inhibition. Hopefully we know that in an enzyme reaction that is labelled as having end product inhibition, it's where there's a system in place to stop too much product from building up. In this case, it's that it helps to prevent too much ATP being produced. So now we have to think about how do we go about explaining how this happens. So we need to talk about inhibition of the enzyme, but also it's explained, so we have to give a reason why. So if this stage slows down, why would that reduce the amount of ATP produced? So if we don't have this stage happening, this decarboxylation and this production of the NADH H+, then the cycle's not going to continue. We're not going to end up then being able to do it again to get the 5. We're not going to be able to produce the 5 carbon molecule. So we can't continue the cycle at that stage. You could also link to the idea that then obviously if the cycle doesn't continue, we don't have enough coenzymes going to the electron transport chain to produce enough ATP as well. How? So that's that one stage. That's the why. Why does this show that it produces less ATP? And that's what we want from end product inhibition. It shows that we're stopping too much ATP building up. So why is that? How would it stop the enzyme? How would end product inhibition stop it? So it would have to be through the non-competitive inhibition of the enzyme, and ATP would have to do that. So to get full marks here, we have to explain what ATP would do to the enzyme, how that would stop it slowing down, how end product inhibition works, and how that would regulate the amount of ATP being produced. So why would that stop ATP being produced until it was used up again, and then we'd go again. So the first mark is going to come from saying when there's a lot of ATP, it's going to bind to the allosteric site of IDH3, or the enzyme. You don't have to specify IDH3, you can say the enzyme and still get the marks here. But we would need to make it clear that it's non-competitive inhibition, so it's binding away from the active site. What does it do to the enzyme? It causes the shape of the active site to change shape. How does that prevent more ATP from being made? Well, it means the 5 carbon molecule is not produced, so the cycle cannot continue. Then we could talk about if we wanted to, less ATP is being produced, so the concentration of ATP would then decrease as it's being used up until it's no longer inhibiting, but I've not put that mark in my answer because it's maximum 4 and there's 5 options. I've just then summarised why this is then an example of end product inhibition. It's because it reduces the amount of ATP produced to stop it building up or to stop too much being made at once. The image below shows a cross-section of 2 blood vessels taken under a light microscope. State what type of vessel is represented by each letter and explain your answer using evidence from the diagram. So when it says type of vessel, it means is it an artery, a vein, or a capillary? And it's asking us to give a reason for our choice, but using evidence from the diagram. So not just a reason why we think it looks like that and just giving ideas about what the structure of the two vessels looks like. We have to be able to show it's something that we can see from the diagram. So for A, I can see that it's got a wider lumen than B. B, I can see that it's got a thicker muscle wall than A. I can also see the folded, kind of wrinkled endothelium on the inside of B. And then obviously you can do the opposite. So narrower lumen for B and then thinner walls for A. So notice I've used like comparative, so thicker, thinner, because that's what we're going to have to use to, we're basically comparing the two to decide which one is which. So A must be a vein because it has a wider lumen. Okay, so just be sort of clear about that comparative element to your answer, because, or you can say the widest lumen of the two. And obviously we are kind of assuming neither of these are capillaries because we can't see kind of a one cell thick wall here. B therefore must be an artery because it has a thicker muscle wall, or you could have said it has the narrower lumen. But you've got options in the mark scheme you can see there, but those are the ones I chose. But as long as you've got A is a vein and B is an artery, and you've got a matching reason that makes sense, that is something you can see from the diagram. For example, you couldn't put A is a vein because it has valves, because you can't see a valve here. So you have to make sure it's something you can see from the diagram and that it matches the correct blood vessel and that you've got the blood vessels the right way around, and then you get the 2 marks. Hemotoxylin and eosin stain were used on the tissue before it was viewed under the microscope. Explain the purpose of using 2 different stains. So we need to explain why you would use 2 different stains, and specifically because it's 2 marks, what do the 2 different stains actually help with? Why would you stain, but why would you use 2 different stains? So they can help us to see detail. So staining helps to see detail that you would otherwise not be able to see because it would be transparent, for example. And the fact of using 2 different stains provides a contrast, which helps you to identify and see different structures from each other, like the nucleus and the cytoplasm. So we need to say, using 2 different stains helps to see detail. And the 2 stains or 2 different stains can help to recognize different structures, for example, the nucleus and the cytoplasm. Here, really, it's about understanding, and this question gets asked, a lot, because we have to understand and be able to explain the purpose of staining and differential staining. So it's always going to be about the fact that if we don't stain things, we can't see the detail because it would be transparent. And the more stains you use that can stain different tissues or structures within the cell, then the more detail you can see, because you can actually start identifying different structures from each other. After carrying out a heart dissection, students wanted to investigate the physical properties of arteries and veins. They cut rings of artery and vein tissue and then set up some equipment as shown in the diagram. So we've got a hook, we've got a ring of artery or vein tissue, and it's been attached to a mass carrier and some masses have been added. And we've got a ruler behind it so we can measure the length of the ring of tissue. When it's talking about measuring the physical properties of arteries and veins, this is going to be about strength, elasticity, so the different structures enable them to have those different properties that we know arteries and veins have, so their structure matches their function. This is to look and investigate and kind of compare the differences between those 2 tissues. Give 2 safety precautions that students must use when collecting and handling the vessel material. Okay, so this is a PAG skills related to dissection, and the skills we've got here is thinking like handling biological material, and then also thinking about kind of cutting, and actually it says collecting and handling. So we've got this idea of what should you do when you're handling this tissue, how should you be safe, but also when you're collecting it, so they must have cut the rings of tissue, it says in the start of the question. So our cutting skills, scalpel use skills, how to be safe when we do that. So the main 2 that spring to mind for me here was to wash your hands after you've touched and handled the material is a safety point. Cutting safely, so cutting away from you or cutting onto an appropriate surface, not into your hand, things like that. or the idea of kind of disposal of material or cleaning up and things after using, obviously, material that is potentially a biohazard. So any of those, and you've got the mark scheme to kind of give you the more detail that you can pick. We've just got to pick 2. So washing hands after handling the tissue is going to be my first one. Again, you have to be specifying that it's after handling the tissue. You can't just put wash your hands. That's not an effective and descriptive enough answer to get the mark here. And then cutting away from the body when slicing or cutting the vessel tissue. The results are shown in the table below. So we've got some rows for the artery, we've got some rows for the vein. The masses added were the same for each one. We've got the original length with the mass carrier, and then we've got the new length with the masses. So every time we added another mass, we'd measure it with the mass carrier, and then once the masses were added. Calculate the difference in percentage length increase of the original piece of tissue between the artery and the vein by the end of the experiment. So it wants us to calculate the percentage length increase from the start to the finish of the experiment for the artery and the vein. So from 4 to 5.2 centimetres for the artery and 4.5 to 5.1 centimetres for the vein. Calculate that percentage length increase and then calculate the difference in that increase between the artery and the vein. So the percentage increase is going to be the increase in length divided by the start length times 100. So for the artery, that's 5.2, because that's the final one, minus 4, which gives us the difference or the increase, divided by 4 times 100 gives us 30% increase in length. And then for the vein, it's 5.1 minus the 4.5 divided by 4.5 times 100 gives us 8.51% increase in length. So the vein obviously increased in length less than the artery. So the difference between the two percentage increases is 21.49%. And if you round it up to 21.5, then you still get the mark here. Explain the difference in the results between the artery and the vein. So it's asking us to give reasons for the difference in the results, i.e. the difference in the change in length, because we have just calculated that. But you can also get other things from the results table you could use for this as well. So my main thing I'm trying to think of a reason for is, why does the artery stretch more? Why is it an extended length as masses are added? Why does it stretch more with the same amount of mass? Well, what can I link to the structure of arteries? The arteries are more elastic, right? This shows they must be more stretched, able to be stretched more, so they have more elastic tissue. We should know that. So then we need to link the idea of, why do they have more elastic tissue? Well, this is because they need to stretch and recoil to maintain high blood pressure. So this is really a structure and function question about arteries. It's shown you that they've stretched more, they've got longer when the same amount of mass is added. You're trying to come up with a reason why. So the difference between arteries and veins is that the arteries stretch longer. The physical difference between arteries and veins is that arteries have more elastic tissue. The reason arteries have more elastic tissue is because they have to stretch and recoil to maintain high blood pressure, which obviously veins don't need to do. The artery stretched more than the vein because it contains more elastic tissue. So with one sentence there, I've kind of got 2 marks because I've talked about the difference that we've seen, and then I've given a reason, one reason for the difference, and then I've got to say why. This is because the artery has to stretch and recoil to help maintain high blood pressure. The students continued to add masses, and they found the vein tissue broke at a lower mass added than the artery. Suggest why. So this is a suggest why question. They've told you something's happened and you need to use your own knowledge to come up with a reason. So this is showing us that the artery is able to withstand a stronger force than the vein. So we can link that again back to high blood pressure that it needs to withstand. But why can it do that? And think about a structural thing again, because that's obviously what this question is focusing on. It's got more collagen. So more collagen in an artery than in a vein, and that gives it this high mechanical strength, because we know that that's a property of the protein collagen. So arteries have a thicker layer of collagen. Again, I've tried to say thicker every time I've tried to make it a comparative statement. You can literally say arteries have a thicker layer of collagen than veins, if you wanted to withstand high or higher blood pressure than veins. Again, just making it clear that we are comparing the two. And if you put thicker, or you've got this idea of showing that it is a comparative statement, then you can get the marks there. So it gives them a greater mechanical strength. And obviously that means it takes more force to break them. So we've got these two questions that are about a set of results that are kind of linked to the heart dissection practical, but really it's about, do you know the structure and function of your blood vessels? Are you able to tell me what the different structures are and how that helps them to carry out their function and the differences between them? Students set up an experiment to investigate the effect of the direction of light on the growth of the stem and roots of cress seedlings over 48 hours. They set up two treatment groups, each with 60 seedlings. Group A was grown for 24 hours in the dark and then 24 hours with a single lamp placed to the left of the growth chamber. Group B was grown for 48 hours in the dark. So we've got no light entirely for Group B, and we've got no light at the start for Group A for half the time, and then the other half of the time they were exposed to a light that was coming from one direction only. The students measured the growth of stems and roots in each seedling in both groups. The results are shown in the table below. State the type of hormone that regulates these plant responses. So clearly we're looking at the response of roots and shoots to light, so we're talking about phototropism. We could also be thinking about gravitropism because they are connected in this way, but this idea that we're thinking about mostly is the fact that it's affecting the growth of the shoots and the root tips. So what group of hormones does that? It's auxins. You can put indoacetic acid in there, IAA, all of those count because they're all correct answers here. State two factors the student should have controlled in this investigation. So it needs to be factors that could have affected plant growth or that could have prevented us from being able to compare the results between the groups. Control values are always things that are kept the same. So it's always good to go and check what's already been mentioned in the method that's been kept the same. So each group had 60 seedlings, so the number of seedlings was kept the same. The time was kept the same, although the kind of conditions changed halfway through. Both of this was measured over 48 hours. And so we've got to start thinking, okay, they're all the things that really we should have controlled. So what else could we think about in terms of the factors that could have affected the growth of these shoots that we need to keep the same? So the first one, and one that is always a good go-to, is temperature. Obviously, rate of photosynthesis is going to, and rate of diffusion is going to increase with temperature, so that can affect the growth rate. So we want to make sure the temperature should be the same. Mineral ions can affect growth because obviously they've got to have these mineral ions available in order to grow properly. The volume of water they were given as well to make sure they weren't water stressed. There's loads and you've got more even on the list in the mark scheme, but any two of those, something that's going to affect the growth of these plants. The start height of the shoots being similar, not necessarily something you have to have, but obviously if you're not going to measure percentage increase from the start size, which they didn't do, then that should be something you should keep the same. So I've gone for temperature and the volume of water. Again, be specific when you're saying water, don't just say water, you should say the volume of water that each set of seedlings was given. The students wanted to carry out a statistical test to see if there was a difference in the stem lengths between seedlings in group A and B. State which statistical test they should use and give a reason for your answer. So this is saying what test should they use and why for 2 marks, one about the test and one for the reason. So let's look at the data and just remind ourselves they're looking at the difference in the mean length between the stems in group A and group B. So they're looking for a difference between two means. That must mean we're doing a t-test. Okay, so t-test is always going to be the difference between two means. Correlation coefficient is when you're looking for a relationship between two variables. And chi-squared is when it's categoric data. Now the length here, we're measuring length. That is obviously continuous data. It's not discrete. It's not categoric. You're measuring it with a ruler. So clearly that is a t-test because we're looking at the difference between two means. We've ruled out chi-squared. You can rule out correlation coefficient. So we need a mark for saying t-test and a mark for saying the reason why we're using a t-test, and it's because we're testing the difference between two mean values. For plants in Group A, the students also measured the direction of the growth by counting the number of stems and roots that fell into the following categories. So we've got towards the light, away from the light, and not obviously towards or away from the light, so no direction. So remember, Group A is the one that had the light coming in one direction. It wants us to describe and explain the results in the table. So what patterns can you see? What differences can you see between the stems and the roots and the towards and away from the light? And give some reasons for this. So reasons why we see these differences in the result. So more stems grew towards the light than roots, and more roots grew away from the light than stems. So that's the easiest one because that's an obvious positive phototropic response and a negative phototropic response. So we can explain that using our knowledge of how IAA works. It appears to be that more stems responded to the light than, so in terms of like the number of stems that responded to the light overall, more stems did than roots did. But the fact that there were so many or a high proportion of roots that didn't seem to respond to the light at all could be the result of the fact that we're doing it in unnatural conditions. Could have been some variation in some other factor that could have affected the growth of the roots. It could also be that there was only 24 hours that they were exposed to the light in order to measure the growth. And so that time may not have been enough for the roots to actually have had a phototropic response compared to the shoots. So we have to give a reason for everything as well as describing each row of the table. So I would do it like that systematically. And that's what I've done with my answer. So more stems grew towards the light than roots. This is because of the positive phototropic response. IAA will move to the shaded part of the stem and cause more cell elongation on this side, which causes the shoot tips to bend towards the light source. So I've described and explained for the first row. The opposite was seen in roots, where more roots grew away from the light than shoots. This is because of the negative phototropic response. IAA moves to the shaded side of the roots but inhibits growth on this side, causing the unshaded side cells to elongate more, bending the roots away from the light source. So I've described and explained the second row. There were some shoots that did not show obvious directional growth response to the light. This could have been due to the unnatural conditions of the experiment or the short experiment time, as 24 hours in the light may not have been enough time to see a response in roots. There are two carrier proteins, one which actively transports auxins into cells and another that facilitates their diffusion out of cells. Synthetic auxins have been created which are used as weed killers. They have a shape that allows them to be actively transported into the cell using a carrier protein, but they're not able to use any channel proteins or cross the cell membrane. Explain how these synthetic auxins could cause a plant to die. So we've been told that it's these auxin can actively be transported in, and then there's a way, normally maybe a channel protein or something, that enables diffusion out. So not necessarily simple diffusion, but facilitated diffusion out of the cell. So we've been told that this synthetic auxin can get in, but it can't leave. And so therefore it's going to accumulate or build up within the cell. If something can get into a cell but can't get out again, then it's obviously going to build up. How does it cause the plant to die? Hopefully we know this about synthetic auxins as weed killers, or weed killers that are containing auxins, then we know how they work. Basically, they cause the plant to grow too quickly and cause it to collapse then. So it grows up too tall without the ability to reinforce those cell walls and get some thickening, maybe lignin and things like that in order to prevent it from falling over. So we need to be able to link those two bits of knowledge together. So we have to say that it can't leave the cells, so it will accumulate. And that's the bit that we've interpreted from being given the information in the question. High concentration causes rapid cell elongation, which causes the plant or the stem to grow too quickly and collapse. And then obviously if the majority of your plant is lying down on the floor collapsed, it it's not going to be doing photosynthesis as much because it's not standing upright. It's probably going to be shaded by other plants, and that's how it causes it to die.
Ouch! This is why in some videos I'll explain scratches.